Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Students, today we are going to study proteins in detail. Today we will take a detailed account means complete tutorial from federal biology for class 11. Well, this is chapter number 2 and we are going to study proteins. So, what are proteins? Proteins are the main structural components of the cell. Proteins are the main structural components of the cell. So, what are the main structural components of the cell? These are the proteins. All proteins contain carbon, hydrogen, oxygen, and nitrogen, while some contain phosphorus, sulfur. Few proteins have iron, iodine, and magnesium incorporated into the molecule. So, all proteins contain four basic elements, four compulsory elements: carbon, hydrogen, oxygen, and nitrogen, while some contain phosphorus and sulfur. Similarly, there are few proteins that have iron, iodine, and magnesium incorporated into the molecule. Now we study structure of proteins. Chemically, proteins can be defined as polymers of amino acids or polypeptide chains. So, what are these? These are polymers of amino acids. Chemically, proteins can be defined as polymers of amino acids. So, chemically, proteins are polymers of amino acids or polypeptide chains. A protein may consist of a single polypeptide or a more than one polypeptide. So there are number of proteins. So proteins may consist of a single polypeptide or more than one polypeptide chains. Uh, as we studied that proteins are polymers of amino acids, so now we will have a look at amino acids. What are amino acids? Amino acids are the building blocks of proteins. Amino acids are the building blocks of proteins. Uh, Amino acids are the monomers that are linked together through peptide bond to form proteins. There are many amino acids known to occur, but only 20 are commonly found in proteins. There are many amino acids that are known to occur, but only 20 are commonly found in proteins. The amino acids are built on a common plan, so all amino acids share a common structure. Well, they are built on a common plan. Each contains a carbon atom. It is called alpha carbon. To this, a hydrogen atom, an amino group, a carboxyl group, and a variable group known as R group are attached. So every amino acid contains a carbon atom that is called alpha carbon, and to this amino group, carboxyl group, hydrogen, and a side chain are attached. The R group has a different structure in each of the 20 biologically important amino acids. So when we talk about amino acids, there is R group that is known as side chain. So the R group has a different structure in each of the 20 biologically important amino acids. The R group has a different structure in each of the 20 biologically important amino acids and determines their individual chemical properties. And so this is the group, R group, that determines uh, their individual chemical properties. It determines the individual chemical properties of amino acids. Two simplest amino acids, glycine and alanine, are shown in figure 2.21. So there are two amino acids that are shown here. Here we can say that here if we say that there is R present, So uh, one minute. Sorry. Here we can see this is glycine. Again, alpha carbon amino group. This is carboxyl group, and this is hydrogen atom, and this is R group. This is glycine. And if this R group contains methyl group, this is called alanine. So these are shown here. And now we come dipeptides and polypeptides. Dipeptides and polypeptides are formed by the condensation of amino acids on the ribosome under the instructions of a messenger RNA, which takes these instructions from DNA. So how dipeptides and polypeptides are formed? These are formed by the condensation of amino acids. So dipeptides and polypeptides are formed by the condensation of amino acids, and this condensation takes place on the ribosomes. Uh, this condensation takes place on the ribosomes under the instructions of messenger RNA. It takes place under the instructions of messenger RNA, which takes these instructions from DNA. And this messenger RNA is a transcript of DNA, and it takes these instructions from DNA in the nucleus. This process is known as translation, and this process is known as translation. During this process, when an amino acid reacts with another amino acid, the hydroxyl group from carboxylic acid group of one amino acid and a hydrogen atom from amino group of other amino acid are liberated and form a water molecule. As a result, a bond is established between carbon of carboxylic acid group and nitrogen of amino group of two amino acids called a peptide bond. So what happens here? We can see this is glycine, this is alanine. So these are two amino acids. So when they react, a condensation reaction takes place, and a hydroxyl from carboxylic, a hydroxyl group from carboxylic carbon, carboxylic group, and hydrogen atom from amino group combine, and they release a water molecule. And as a result of the removal of hydroxyl and hydrogen atoms, a chemical bond is formed between the carbon of carboxylic acid group and nitrogen of amino group. And this bond is called peptide bond. Hence, a product of two amino acids is formed, which is known as dipeptide. And because this product consists of two amino acids, it is formed by the condensation of two amino acids. It is called dipeptide, and it is formed due to bond formation. And this bond is known as peptide bond. And this dipeptide is known as glycyl alanine. A dipeptide has two ends. One is called amino or N-terminal end, while other is called carboxylic acid or C-terminal end. Well, there were amino and carboxylic acid terminal or end. These are present in glycine. These are present in alanine. So when a dipeptide is formed, again an amino group is present. Again, a carboxylic acid group is present. So again, both groups are available for further chemical activity, for further chemical reactions, to form to link more amino acids and form tripeptide, tetrapeptide, polypeptides, and proteins. So our dipeptide has two ends. Here we see that our dipeptide has two ends. One is called amino or N-terminal, one is called amino or N-terminal, and while other is called carboxylic acid or C-terminal end. A new amino acid can be added in this chain from its carboxylic acid or C-terminal end in the same way. So a new amino acid can be added. Thus, a tripeptide, a product of three amino acids, is formed, and another water molecule is also released. The same process that happened during dipeptide. So when a third amino acid comes, a third amino acid is added, same steps take place, same mechanism happens. Similarly, when several amino acids are linked together by many peptide bonds, the polypeptide chain is formed. So in the same way, in the same manner, likewise, when several amino acids are linked together by many peptide bonds, the polypeptide chain is formed. So there are several means many, numerous, certain number of amino acids combined or linked together or bonded together by peptide bonds, many peptide bonds, the polypeptide chain is formed. Now we study structural conformations in proteins. A linear polypeptide with a specific sequence and number of amino acids is called primary structure. There is a linear polypeptide and it has a specific sequence and a specific number of amino acids. So it is called primary structure. So it has a specific sequence and number of amino acids. So when a polypeptide, a linear polypeptide, has a specific sequence and number of amino acids, it is called primary structure. It is shown by all proteins at the time of their synthesis on ribosomal surface. So when the translation is taking place, when protein synthesis is taking place, during the process of translation in protein synthesis, a primary structure is formed on ribosomal surface, and it is shown by all proteins at that time when they are being synthesized on ribosomal surface. After synthesis, a protein does not remain in its primary structure but can be changed into some other structural conformations, particular form, shape, or structure. So, this primary structure after its synthesis is changed into some other structural conformation, some regular configuration, and that is known as particular form, shape, or structure, and that is known as native protein structure. A helical, alpha helix, or flattened sheets. Helical structure is known as alpha helix, or if these are flattened sheets, these are known as beta plated sheets like structure, which are established by hydrogen bonding between opposite charge pairing groups of different amino acids, are called secondary structures. So these are known as secondary structures. This is a chain, a linear chain with a specific sequence and number of amino acids in this polypeptide chain. This is primary protein structure, a sequence of a chain of amino acids. And when it is configured or conformed, or configuration or conformation takes place, well, there is secondary protein structure which may be a alpha helix or a beta plated sheet. So when this occurs, it occurs when the sequence of amino acids are linked by hydrogen bonds. It occurs when the sequence of amino acids are linked by hydrogen bonds. So in some proteins, the linear polypeptide is changed into alpha helix, then alpha helix fold again and again by ionic bonds and disulfide bridges to form a globular shaped structure, the tertiary structure. So what happens in some proteins? The linear polypeptide that was changed to alpha helix. This alpha helix folds again and again by ionic bonds and disulfide bridges to form a globular shaped structure that is known as the tertiary structure. So here we can see this alpha helix is present here, and it is being formed. It is being arranged into another type of structure, the next type of structure, the next level of structure, and it is known as tertiary protein structure, tertiary structure of proteins. It occurs when certain attractions are present between alpha helices and plated sheets. So what are the interactions? These are the ionic bonds and disulfide bridges. Similarly, some proteins exist in very complex structures in which more than one globule is attached together by hydrophobic interactions. Such structures are called quaternary structures. So sometimes there is a complex structure, there are two or more polypeptide chains, and these are arranged together by hydrophobic interactions to form a globular structure, and this structure is known as quaternary protein structure. Is the protein consisting of more than one amino acid chain? So there are more than one amino acid chains. There are more than one polypeptide chains. Means there are two, at least two or more than two polypeptide chains present. And when these are folded together to form a globular structure by hydrophobic interactions, this type of structure is known as quaternary structure. Significance of amino acid sequence. Sequence of amino acid in a polypeptide is a characteristic feature of primary structure of proteins, which is responsible for proper functioning of protein. Amino acids have a specific sequence and a number. They are present in a specific sequence and number in a polypeptide chain. So this specific sequence and number of amino acids in a polypeptide chain is a characteristic feature of primary structure of proteins, and this is responsible for proper functioning of proteins. It is determined by the sequence of nucleotides and DNA. We already have studied that there is central dogma in the cell in which DNA uh, transfers its information to messenger RNA, and the messenger RNA moves to cytoplasms where it helps other metabolic machinery, other metabolic components to synthesize polypeptide chains on ribosomal surface. So, uh, in short, we can say that this polypeptide chain is the structure, the sequence, the folding is determined by the sequence of nucleotides and DNA. Even due to point mutation, what is point mutation? Change of single or few nucleotides and DNA. The sequence of amino acids in a particular protein may be disturbed, which causes wear defects in the body as it happens in sickle cell anemia or heart disease. So here we see that there are mutations maybe on smaller scale, maybe on larger scale. So there is point mutation, change of single or few nucleotides in DNA. And if any point mutation occurs, it disturbs the structure and function of the whole polypeptide of the whole protein. So it is exemplified by the this disease, sickle cell anemia. Here we see it in detail. Normal red blood cells are disc shaped and look like duff nuts without holes in the center. So normal red blood cells are disc shaped and they look like duff nuts without holes in the center. They move easily through our blood vessels. Red blood cells contain an iron-rich protein called hemoglobin. This protein carries oxygen from the lungs to the rest of the body. So red blood cells are disc-shaped. They move easily through our blood vessels and they contain an iron-rich protein called hemoglobin. This protein carries oxygen from the lungs to the rest of the body. Normal hemoglobin, this is shown hemoglobin A, contains four polypeptides. It is composed of, it is made up of four polypeptides. Two alpha chains, which consist of 141 amino acids in each chain, and two beta chains, which consist of 146 amino acids each. So there are four polypeptide chains, two alpha chains, and each alpha chain consists of 141 amino acids, and there are two beta chains, and each beta chain consists of 146 amino acids. Sickle cell anemia involves sickle or crescent shaped red blood cells. So there is a serious disorder that is known as sickle cell anemia. It is a serious disorder, and what happens during this disorder, in which the body makes sickle or crescent shaped red blood cells. Sickle cells contain abnormal hemoglobin called sickle hemoglobin. So there is abnormal hemoglobin, sickle hemoglobin. Sickle hemoglobin causes the cells to develop a sick or crescent shape. So, so what happens? These sickle hemoglobin cells become a sickle or crescent shaped. Sickle cells are stiff and sticky. So they are stiff, they are sticky, means we studied that normal red blood cells are disc shaped. So their structure is distorted, has been disturbed, has been distorted. They tend to block blood flow in the blood vessels of the limbs and organs. Blocked blood flow can cause pain and organ damage. So they tend to block blood flow, and this blocked blood flow causes pain and organ damage. Sickle cell anemia is caused by a point mutation in beta globin gene in which only one nucleotide is replaced by another, which causes a change in amino acid sequence of beta chain of hemoglobin. So, how this sickle cell anemia occurs? It is caused by a point mutation in beta globin gene. It is occurred by a point mutation in beta globin gene in which only one nucleotide is replaced by another. So here we see this is normal amino acid sequence in normal red blood cells in normal hemoglobin. Here glutamate is present at number 6. When we see sickle red blood cells, there is a single change in amino acid sequence, and this glutamate is replaced by valine, and it uh, different amino acids are being formed. Why? Due to a change in single nucleotide in only one nucleotide. And what happens in which only one nucleotide is replaced by another in which only one nucleotide is replaced by another, which causes a change in amino acid sequence of beta chain of hemoglobin. So only one nucleotide is changed, and a whole new amino acid is formed in its place, and the structure of the red blood cells is disturbed. It becomes crescent or sickle shaped. Sickle cell hemoglobin shows only one difference from normal hemoglobin. Glutamic acid is replaced by valine at position number 6 in beta chain, and the whole functioning, the whole structure is disturbed, is distorted, is affected badly. The next topic is classification of proteins based upon structure and shape. Proteins can be classified into two groups, fibrous and globular. So, based upon structure and shape, proteins can be classified into two groups. So, proteins can be classified into two groups on the basis of structure and shape, and these two groups are fibrous proteins and globular proteins. What are fibrous proteins? These proteins have fiber or filament like shape. These proteins have fiber or filament like shape, so these are called fibrous proteins. Therefore, they exist in secondary structure during function. These proteins have secondary structure. These proteins are insoluble in aqueous medium, elastic in nature, and cannot be crystallized. So these are insoluble in aqueous medium. These are elastic in nature and cannot be crystallized. Examples are collagen, fibrinogen, actin, myosin, and keratin. What are globular proteins? These proteins have spherical or globular like shape. These have spherical shape or globules like shape. Therefore, they exist in tertiary or quaternary structure during function. So they exist in tertiary or quaternary structure during function. These proteins are soluble in aqueous medium, inelastic in nature, and can be crystallized. So these are soluble in aqueous medium. These are inelastic in nature and can be crystallized. So these are completely opposite to fibrous proteins. Examples are enzymes, hormones, antibodies, channel proteins. And in the last, we will study importance of proteins, significance of proteins, functions of proteins, role of proteins. So what is the role of proteins? Proteins are very important molecules in our cells. They are involved in virtually all cell functions. So proteins are very important molecules in our cells. Why? Because virtually they are involved in all cell functions. Each protein within the body has a specific function. Each protein within the body has a specific function. Some proteins are involved in support or composition of body or organ parts. Means they have structural roles. They are involved in sport or of composition of body parts. So they have structural roles, while others are involved in various physiological activities like bodily movement or in defense against germs, while others are involved in various physiological activities like bodily movement or defense against germs. So these are functional roles. Uh, a list of several types of proteins and their functions is given table 2.4 and 2.5. So first we have list of structural proteins. Collagen, it establishes the matrix of bones and cartilages. So collagen, what it forms, it establishes, it forms the matrix of bone and cartilages, means it is present in the matrix of bones and cartilages. Elastin provides support for connective tissues such as tendons and ligaments. So it provides support for connective tissues such as tendons and ligaments. Keratin, it strengthens protective coverings such as hair, nails, quills, feathers, horns, and beaks. Stone, it arranges the DNA into the chromosome. So it arranges the DNA into the chromosome. Next is the list of functional proteins. There are enzymes. The most of enzymes are proteins which control metabolism. Most of enzymes are proteins which control metabolism. They speed up the biochemical reactions. Next are hormones. Some hormones are protein in nature, which are involved in the regulation of physiological activities such as regulation of glucose level, calcium level, digestion, blood pressure. So there are different physiological activities. So there are hormones which are protein in nature, and these are involved in the regulation of these physiological activities. Similarly, another type of functional proteins are antibodies. These proteins are produced by white blood cells in response to antigen, a foreign particle, and provide immunity. So these proteins are produced by white blood cells. These are produced in response to antigen, and these provide immunity. Next is hemoglobin. It is found in red blood cells and is involved in the transport of oxygen mainly, and carbon dioxide to some extent. Next is fibrinogen. It is found in blood plasma and is involved in blood clotting process. So fibrinogen is involved in blood clotting process, and it is found in blood plasma. Ova, albumin, and casein. Ova albumin is found in egg whites, and casein is a milk-based protein. Both of them are involved in the storage of amino acids. So today we took a detailed account, detailed session on proteins from chapter 2 of federal biology for class 11. So it's over for today. It's and for today. Hope you will study it and will try to have any questions if you have. So we will try to answer you. Thank you. Stay blessed. Allah Hafiz.